membrane. Okay. Tonicity, which is on page 90 and 91 of your textbook. This one is pretty simple to understand. I think you guys are going to be able to really understand this one, but it seems like students have a hard time understanding osmosis. These fall under the category of osmosis as well. Tonicity, ability of a surrounding solution to affect fluid volume and pressure in a cell, depends on concentration of what non-permeating solutes. So it's associated with osmosis. So when you have a hypotonic solution, a hypotonic solution simply means that you're going to have hypotonic solution. I'm thinking how I can explain this to make it more user-friendly. A hypotonic solution, okay. You're going to have a higher concentration of water in the extracellular fluid, but a lower concentration of the other molecule. Let's say, for example, okay, we're going to take sodium chloride. I'm about to talk about saline. Outside of your red blood cells, or yeah, you see your red blood cells on page 91. We're going to use that example. A cell that's hypotonic, outside in the extracellular fluid, we know what extracellular fluid is because we've already talked about it and it's on your exam. So outside of your cell and your extracellular fluid, let's say you have 10% sodium chloride, but you're going to have 90% water. Okay? So hypotonic, you're going to have less of that sodium chloride outside of your cell. Hypo means less or below, compared to the intracellular fluid. Now, in the intracellular fluid, you're going to have 10% water, but you're going to have 90% sodium chloride. Okay, remember this. What do you have in the extracellular fluid? 10% sodium chloride, 90% water. What do you have inside of the cell, the red blood cell? 90% sodium chloride, 10% water. You see how they've reversed? So what's going to move? Water. Because you've got 90% water in the extracellular fluid and you only have 10% in the intracellular fluid, water is going to move from high to low across the selectively permeable membrane, and it's going to cause the cell to swell. All right? The cell will swell. That's pretty simple to understand. Now with hypertonic, your cell is going to lose water. All right? It's the opposite. So with hypertonic, you have in the extracellular fluid, okay, you're going to have 90% sodium chloride and only 10% water. So you have less water, but you do have a higher concentration of a higher percentage of sodium chloride outside of your cell. However, in the intracellular fluid, you're going to have 90% water and 10% sodium chloride. Inside, 90% water, outside 10% water. Water is going to move from the intracellular fluid from an area of high concentration across the membrane of your red blood cells to the extracellular fluid where there's only 10% water, a lower concentration. The cell is going to shrink or shrivel up. Our red blood cells crenate. Okay? If they can actually, cells can desiccate. Desiccate simply means you're drawing something out of that cell. You're drawing water out of it. So it's going to what? Desiccate. Isotonic, very user-friendly. Causes no change in cell volume. So isotonic, you got 10% sodium chloride outside of the cell, 90% water, Inside the intracellular fluid, 10% sodium chloride, 90% water. You've got a balance there. What's going to happen? Absolutely nothing. Okay? Our red blood cells are in an isotonic solution. Okay, isotonic solution. Okay, concentrations of non-permeating solutes in bath and ICF, intracellular, are the same. That in bath means the extracellular fluid. And the ICF are the same. Normal saline, 0.9. Oh, 0 looks weird. 0.9% sodium chloride is an example. Okay, 0.9% sodium chloride, guys, is normal saline. All right, they're used in when people are dehydrated. Normal saline, of course, is put in IV drips. It's all it is sodium chloride. It's an electrolyte replacement because when you're dehydrated or you have a severe case of diarrhea, you're going to lose a lot of electrolytes. Okay, so it's a source of water and electrolytes. It's going to replace electrolytes. It's used to supply water and salt to the body, and it's going to maintain a balance of osmotic pressure between the body's cells and the fluids. Okay, so you need that 0.9%. Well, 5% saline keeps our body in homeostasis or within balance versus pure water. If you put pure water in an IV drip, okay, and it goes into the cell, that cell is going to pop. Okay, if it's too concentrated, the solution, that's when the cell is going to shrink or it's going to desiccate. That's why 0.9% is so important when it comes to a lot of medical issues. Let me just put it that way. All right. So hypotonic, okay, because you have a higher concentration of water in the extracellular fluid, it's going to move into the cell, which only has a lower concentration of water, and the cell is going to swell or it's going to burst. In a hypertonic solution, because you have more water inside of your cell, more water inside of the cell, and less water in the extracellular fluid. So the water is going to flow out of the cell into its surrounding environment, which is your extracellular fluid, and it's going to cause the cell to shrivel up or your red blood cells to crenate. Simple as that. All right. When your plants lose water, okay, or they're gonna kind of wilt or shrivel up, okay, that's a hypertonic. You need to water your plants. When you give your plants water, they all of a sudden plump up. They become hypertonic. They're not going to burst because plants have a cell wall. We don't. Our cells can burst. All right. And also there's that balance going on there as well, which means your plants are now in that isotonic state. So when plants shrink because they're not, or wilt because they're not given enough water, it's called plasmolysis. Plasmolysis. That's talked about in Biology 103. Okay. And then when you have enough water in that plant and the plasma membrane is going to actually push itself up against the cell wall, and turgor pressure is going to be created. So the pressure, hydrostatic pressure, is going to go up in that plant, but it's not going to burst. So that's isotonic, hypertonic, hypotonic. Hope this helps. Just remember 0.9% normal saline is important, okay, when it comes to keeping our cells in an isotonic state. All right. And here you have it. These are carrier-mediated transport proteins right here. I'm going to talk about, okay, ATP, sodium potassium pump. All right, let me just say this. Ions have to be pumped across. So you're going to see this in chapter 11 here. There's a 3-to-2 ratio of sodium ions versus potassium ions. When we talk about an action potential in chapter 11, we're going to talk about how sodium is supposed to go. Okay, this is weird. I don't think I need to talk about this because this drawing looks kind of weird. Sodium typically will go into the cell and potassium is going to go out. And that's when, but anyway, this is a pump. So it's called a sodium potassium pump because both sodium and potassium are being pumped across this is like a channel protein right here. So these ions that have a positive or a negative charge, they have to be pumped across. Okay. So ATP is needed over here. And ATP, remember, once you lose that last phosphate group, it's going to become adenosine diphosphate and an inorganic phosphate group, and that's when these ions are going to start moving across this protein here. Okay? Half of daily calories utilized for sodium potassium pump. All right. Resting membrane potential, we're going to talk about in chapter 11. We're going to talk about local potential, resting membrane potentials, the resting membrane potential of a muscle. We're going to talk about the resting membrane potential of a nerve cell or a neuron as well, which actively involves the functions of the sodium potassium pump, because it's based solely on the membrane potential has a negative charge. Okay, so it's based solely on the number of negative charges that you're going to have in the intracellular fluid, which will be potassiums and other anions. Anions have a negative charge. Cations have a positive charge. Sodium, because sodium is the only element outside in this extracellular fluid, and it has a positive charge. Look at all of these sodium ions. This is a cation, okay? Now, the inside is a little bit more electronegative because you've got more potassium ions in here and other anions. Anything that has a negative charge is considered an anion, okay? Sodium glucose transporter, of course, is a type of transport protein as well. Vesicular transport, phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis, guys, simply means cell eating. Okay, in the early part of the chapter, we were talking about pseudopods and how, your neutrophils, the amoeba, your macrophages, those white blood cells. Okay, this is how they engulf. Remember, bacteria, or how amoebas get their food. So cell eating, engulfing large particles, that's a form of endocytosis. Endocytosis simply means something is coming into the cell. Phagocytosis, cell eating, pseudopods are involved, phagosomes, those macrophages that we saw on page 88, figure 3.13. With the macrophage that does the complete checkup on our immune system, and you can see it reeling in with the filaments, the bacteria. Pinocytosis, on the other hand, takes in little small droplets of fluid, extracellular fluid often, so it's going to come in. That's cell drinking. Receptor-mediated endocytosis. Now, this one is kind of a little bit different. If you look at page 95, figure 3.2, that's an example of receptor-mediated endocytosis. Okay? So, receptor-mediated endocytosis is highly correlated with hypercholesterolemia. high cholesterol. Okay, we talked about the HDL and the LDL earlier, so now we're about to talk about how the impact, how LDL is removed from the blood. Okay, if you look on page 94 of your textbook, that last sentence, okay, no, the last paragraph, vesicular transport. One example of receptor-mediated endocytosis is the uptake of low-density lipoproteins. Those are the LDLs, remember? These are the ones, the LDLs are the ones that will, actually, that's the bad cholesterol, and they are highly associated with cardiovascular diseases. Okay, but anyway, if you go on down, much of what we know about receptor-mediated endocytosis comes from studies of a hereditary disease called familial hypercholesterolemia, which shows the significance of this process to cardiovascular health. Okay, see deeper insight 3.3. That's that clinical application that's on your study guide. Okay, so here's how it works. If you look on page 95, receptor-mediated endocytosis. Okay, if you look at the, let me just find the picture. This right here. All right, here's the plasma membrane or your cell membrane. These are receptors right here. Okay, now we're going to say this is on our blood vessels right here. These are the little tiny molecules that have to bind to these receptors on the membrane. Once they bind to that membrane, it's called an invagination, and you have to have this coated pit that's going to form. And this is clathrin right here. Clathrin is needed to kind of hold these receptors in place. So plasma membrane is going to sink inward. You see the little hole or groove that's forming, and it's going to form this little clathrin coated pit right here. And then all of a sudden, the membrane is going to just fold itself around these little particles which are on these receptors, and they're going to take them, bring them into the cell, okay, containing concentrated molecules. The molecules were in the extracellular fluid. Now they're in the ICF. They're inside of what? The cell. So clathrin is like an address label, okay. Now, our blood vessels have these LDL receptors. That's why it says this is associated with receptor-mediated endocytosis. Okay, so with this little pit right here, what happens is the LDL is going to be absorbed into this pit right here, the low-density lipoprotein, and the lipid is going to be metabolized, and that's where it's going to be broken down. People may not have enough of these receptors for the low-density lipoprotein to even bind to it, you know, which is probably one of the reasons high cholesterol runs in the family. It's genetics. But once that LDL binds to these receptors, it's going to be metabolized, or it's going to be broken down inside of the cell, which means you won't have the LDLs floating around in your bloodstream. Okay, so make sure you read receptor-mediated endocytosis. Yeah, that's very important. So that's that. I think I've pretty much covered everything that's going to be on your first exam. Hopefully you guys will listen to these YouTube videos and, you know, just do very well on your test. All right. Thanks. Good night. Okay, let me just talk about what I need to talk